All right, we'll go ahead and get started. Thanks everybody for coming. Today we have Monica Geber.
Geber.
Sorry. It would have been helpful if you kind of told me that. So Monica got her BA at Reed College in math, and then she did an MS and PhD in population biology, first at MS at Oregon State and a PhD at University of Utah. She had a very short postdoc at the University of Utah following her PhD before she started a faculty position at Cornell, where Pat Doak was her first graduate student. She's probably best known for her work on Clarkia and community ecology in the southern Sierra Nevadas. And more recently, and connected to me, she took a student, Aubrey James, who was my first REU, which I had in Massachusetts before I came here. She's working on pollinators in that Clarkia ecosystem. Today she's going to be talking about ecology and evolution of species geographic ranges, which is quite germane to a lot of work we do here in ecology. So I'll just let you take it away.
Well, thank you for inviting me. This is my first time to Alaska, and as I've been telling everybody, it's the only state in the country that I hadn't been to, so I get to check it off my bucket list, even though I need to spend a lot more time here than just the few days that I'm here. But I've been having a lot of fun talking to people, and it's wonderful to see Pat. I haven't seen her since she left Cornell after her PhD. So the work that I want to be talking about is work we've been doing in about the last decade. And before I go any further, I want to acknowledge my collaborators, because this work couldn't have been done without them. Vince Eckhart at Grinnell College. We were in grad school together, and we've been working on this Clarkia system, this plant, for over 20 years now. David Moeller was a PhD student of mine, and he's become a collaborator. He's at the University of Minnesota. Bill Morris is our demographer. He does all of our demographic work. And then Peter Tiffin at Minnesota was involved with the population genetic and ecological genetic work. So, what the organization of my talk will be as follows. I want to tell you a little bit about why one might be interested in geographic range limits, which most of you know perfectly well why one might be interested. I'll go through some of the hypotheses that have been advanced to explain why there are geographic range limits, or why species don't occur beyond their current range limits. I'll give you a little bit of background on my favorite plant, the genus Clarkia, and then I'll tell you about what we've been finding. So why, one of the reasons obviously to be interested in geographic range limits is to try and understand what species will do when environments change, and we're obviously in the midst of a pronounced change in environments. If you think of the geographic range as the physical manifestation of a species' ecological niche, both its biotic and abiotic environment, what's going to happen when the environment changes? If the environment changes, the location of that physical manifestation will change in space. And a species has several different options available to it potentially. It can either move and track the niche. It can evolve to altered conditions that occur where it is presently residing. Or if it can't tolerate those conditions and it can't evolve, it might simply go extinct. So learning about what species capacities for evolving or dealing with a changed niche is of interest. We know that already that a lot of species are moving their ranges, or at least attempting to track the changing environment. This is a study that was published over almost a decade ago of breeding birds in California, in which 4 transects were recensused for breeding birds about 100 years apart. And the authors asked how many of the species had actually moved their breeding bird distribution. Let me get some water. And in this study, there was evidence that 91% of all bird species had shifted their breeding locations to some extent, and they could correlate these shifts to changes in rainfall and precipitation in that geographic area. But the evidence for species range shifts is much less solid for plants. There is considerable evidence that plants are changing their geographic ranges, or at least their elevational ranges, but they appear to be doing so much more slowly than mobile species like birds. So in this particular study, also in California, using this time herbarium specimens from about a century ago and more current specimens, they've asked again how many had shifted their elevational niche or location. And in this case, only 15% of the species out of 4,000 showed evidence of having changed their elevational distribution. And interestingly, though, what they found was that they looked at species in terms of their seed size, and species with smaller seeded species, a larger fraction of them had shifted, and they had shifted more in their distribution elevationally compared to large seeded species. There also appeared to be a difference between introduced species and native species, which I'm not sure why that was the case, but nevertheless, here only 15% of species had changed their niche. So even when people have shown that species ranges are shifting, they often compare the rate of change in geographic distribution to the rate of change in climate, and most species don't quite keep up with the change in climate. And so that means that anything that persists in the current environment is facing novel conditions. And in plants, in fact, we have evidence for some species, at least, that they haven't fully tracked the warming post-Pleistocene, so they haven't really reached their northern range limits to keep track of past warming. So in these cases, species are going to have to evolve in order to adapt to changing conditions. Another reason for being interested in geographic ranges and environmental change is that we know from past history of Pleistocene warming that most species respond individualistically to climate, so you don't get the wholesale marching of an entire community tracking climate change, but each species responds differently. And what that means is that we are producing new communities consisting of different members or different relative abundances of species, and so we're creating new species compositions and potentially new ecosystem functions. From a basic biologist point of view, one of the reason I got interested in geographic ranges is that it had always been hammered into me that what ecologists and evolutionary biologists were interested in or wanted to explain was the distribution and abundance of species. And if you think about the geographic range limit, all this is that on one side where the geographic range occurs, the species is present, and then across the boundary, the range edge, the species is absent. So this is a clear-cut case of presence, absence, or abundance and distribution, and trying to understand why a geographic range edge occurs where it does is trying to get at some of the fundamental questions in ecology and evolutionary biology. So that's how I got started on studying geographic ranges. And I chose to do this, okay, well, let me first get to some of the hypotheses. There are basically four broad classes of hypotheses about why species aren't distributed everywhere or why they aren't distributed beyond their current range edge. And one is simply historical. And what this basically means is that it's just a matter of history. The species hasn't had a chance to arrive at a particular location yet. So this is essentially dispersal limitation as an explanation for why a species is absent from a particular region. The next is it with or without dispersal limitation, it's suggested that the environment essentially deteriorates towards the range edges, starts to fall outside the niche of an organism, and so becomes no longer favorable for that organism. That translates into a demographic limit such that populations at the range edges are less productive or less persistent. And so that explains why populations don't persist beyond the range edge. But then this brings into question just one more hypothesis, and that's, well, why don't they evolve to adapt to conditions, environments beyond the range edge? So these are the four classes of limits that have been proposed. I'm going to focus in my talk on the last three, but I'll show you some evidence that there may have been dispersal limitation in our system. So, just to reiterate, if the environment deteriorates from the center of the geographic range to the range edge, one should be able to quantify changes in some important environmental factor as you move from center to range. This should then translate into a pattern where either populations are larger, more frequent, more abundant at the range center, and then taper off towards the range edge. This could also be reflected in differences in population growth rate, which we might expect then to be highest at the range center, where conditions are most favorable, and lowest at the range edge. And if there are extinctions, we would expect extinctions to be most common at the range edge. Then from an evolutionary point of view, as I said, the range limit presents a paradox, and Ernst Mayr expressed this rather nicely. We know that organisms can evolve, often very rapidly. So why is it that some range limits seem to be very stable? Why don't organisms evolve to move beyond the conditions reflected within their current range? And so some of the predictions that are made from the evolutionary limits is that range edge populations, potentially because they're small or go extinct fairly often, or have been subject to strong selection under these adverse conditions, they lack genetic variation, and that may prevent them from adapting further to conditions beyond the range edge. Other hypothesis is that what's preventing adaptation to conditions at or beyond the range edge is that gene flow will tend to be biased from the larger, more abundant populations at the range edge towards the range center, towards the range edge, introducing alleles from the center to the range edge that are maladapted. So we've been trying to address some of these hypotheses in this group of plants. The one that we focus on is Clarkia xanthina. The genus Clarkia is a genus that is centered on California. It's named after Lewis and Clark. And most of the species occur within California. A few reach into Oregon and Washington, and one species somehow got to Chile. They're all annual plants, and most of them are outcrossing. They depend on bee pollination. but the evolution of self-pollination has evolved 10 or 12 different times independently within the genus. So I had been working on this plant for other reasons, other questions, and when I got interested in geographic ranges, this species seemed appealing because it has a relatively small geographic range, and so I could study it essentially through most of its geographic range. So it's distributed in the southern Sierra Nevada. This is Bakersfield right there. And it reaches its highest abundance in the southern Sierra Nevada, though you can find scattered populations in the transverse ranges. And so I'll tell you what we've been learning about the environmental limits, the demographic limits, and the evolutionary limits. So we've been focusing on its geographic range in the Sierra Nevada, and mostly we've been focusing on the eastern range limit, what's limiting spread along this eastern range limit, because there isn't an abrupt change in habitat or anything, unlike the western range limit, which moves from the mountains down to the Central Valley, which is a radical change in habitat. As you move up the mountains, you're first in grasslands. A little bit higher, we get a mixture of oaks and grasslands and pines. Higher up still, we get into chaparral, shrubby vegetation. And then if you move beyond the eastern range, it becomes drier and drier, and we get into desert habitat with Mojave influences. At lower elevations and at higher elevations, we get into Great Basin type of vegetation. So the so Clarkia occurs through all of these vegetation types. It's always in open areas within those habitats, but there's not an abrupt change in habitat as you get to its eastern range edge. It's a pretty spectacular plant. In a good year, it can cover entire hillsides, and it's very appealing, and it's pollinated by these solitary native bees. In terms of its life cycle, it's a typical winter annual. The seeds germinate in late fall or winter with winter rains. Plants grow slowly during the spring when the temperatures are still cool. The plant flowers in June, sets fruit late June, early July, and then the seeds drop sort of passively from these fruits during the summer and into the fall. And the seeds can germinate immediately the next year or stay dormant in the seed bank for multiple years. So I'll tell you a little bit I'll start with what we've learned about the environment. We've tried to characterize both the abiotic and the biotic environment, certain aspects of the abiotic and biotic environment. What we've been able to describe based on having weather stations scattered throughout this geographic range is that there's a gradient of precipitation as you move west to east, and in particular a decline in growing season rains, spring rains, as you move towards the eastern range edge. In addition, the rains are more variable from year to year as you move towards the eastern range edge. And then it does happen that the eastern range edge does coincide with the geological fault, where the bedrock has been changed from a granitic soils to metasedimentary rock. We don't think that that's actually an absolute limit on the species' geographic ranges, because right as you get over to the other side of the fault, you're back into granitic soil. So if they could just make it across that metasedimentary rock, they should be able to find granitic soils, which they prefer. when you look at the effect of these abiotic factors on plant performance, what we find is consistently plants decrease in size from west to east. So as you approach the range limit, they are much smaller. These are data looking at plant height as you move from west to the eastern range edge in 2 different years. That's accompanied by changes in plant water potential. So midday water potential are much lower as you get towards the range edge. We've also found that the metasedimentary rock makes it more difficult for plants to access water. So for a given soil water content, the soil water potential is much lower for metasedimentary rock, so probably making it harder for plants. And we have done species distribution modeling or niche modeling to look at the importance of these various abiotic factors on the distribution of this species, and we can show in these models that there's a fairly high sensitivity to both temperature, precipitation, variation in precipitation, and also bedrock. And our models do predict fairly accurately the actual distribution of the species. What's kind of cool about Clarkia is that as with other members in the evening primrose family, they have very large pollen grains that are sort of stuck together with these sticky threads, viscid threads. They are pollinated by, most importantly, by a diverse group of solitary bees, about 10 species throughout the genus of Clarkia. These are solitary bees that have evolved to specialize on Clarkia, so they only visit Clarkia for pollen and nectar. Some of them have actually evolved specialized hairs that are particularly good at handling this large, sticky pollen grains. We've characterized the pollination environment for Clarkia xanthina across its range by following pollinator visitation rates to plants over multiple years across multiple sites to see if there's any geographic pattern to pollinator visitation rates, and also to ask whether plants are limited in how much pollen they're getting. Are they getting enough visitation and enough pollen for reproduction, and does this vary geographically? And what we find, this is where the PC doesn't reproduce faithfully my slides, but the basic pattern, as you can see, we're plotting easting again on the y-axis, so this is longitude from west to east, and plotted on the y-axis is collection rate of pollinators on plants, and the general pattern is repeatedly, as you move across the geographic range towards the eastern range edge, the collection rate of bees, or so the visitation rate of bees, declines. So plants at the range edge seem to be getting fewer visits by pollinators. Then if you ask through experimental manipulation of how much pollen plants get, so we supplement pollen on flowers, on experimental flowers, and compare their seed set to control flowers that are only visited by the native pollinators, to ask how much pollinator limitation is there to reproduction. We find the opposite pattern in that the greatest pollen limitation to reproduction occurs at the eastern range edge relative to the west. So that would suggest pollinators are becoming limiting as you approach the eastern range edge, and we do know that none of the specialist pollinators are found east of the eastern range edge. On the other hand, when we look at herbivory, there are 2 forms of herbivory. The plants are eaten whole or browsed by mammalian herbivores, mule deer, ground squirrels, and the ubiquitous cows. But then there's also fairly severe herbivory on fruits, mostly by a generalist grasshopper, but there's also a seed predator fly that lays its eggs within the fruits. And again, we've done a similar thing to what we did with pollinators. We've quantified levels of herbivory on plants in different populations spread across the range in multiple years. And for both fruit herbivory and plant herbivory, we see a general decline in herbivory rates as you move towards the eastern range edge. So plants at the eastern range edge are not getting pollinated enough. They're not seeing enough pollinators, but they're also getting eaten less. And so these two different sorts of things tend to balance each other out. So the loss of seed due to not being pollinated enough is compensated in part by not losing those seeds to herbivores. So in general, there seems to be a weakening of biotic interactions as you move from east to west, or from west to east. If we now turn to the demographic limits that the plant is faced with, we've asked a number of questions. We've asked first, are populations smaller and less productive at the range edge compared to the center? Do we find more extinctions at the range edge? And are population growth rates lower as you approach the range edge? So for the last 11 years or so, we've been following about 35 populations. We started with a few more, but have gotten to be a little bit more realistic and come down to 35 populations, where at each site we measure the density of fruiting plants every year, quantify how many fruits per plant they have, and how many seeds per fruit they have, so that we can estimate seed production. We've also delimited the area occupied by each population to say something about the size of the population. And what we find is that there is actually no significant variation in population area or area of occupancy of populations across the geographic range. In fact, if anything, there's a slight increase as you move east towards the eastern range edge. The problem with that is that this is a very difficult terrain to move through. This plant likes to be on slopes of about 40 degrees or more, and so it's a little hard to get what is a random sample of populations throughout the range, and it's not quite as steep as you get towards the range edge. So our next move is to try and find a drone that can fly over this terrain and sample the landscape more randomly. Well, there were data points in this figure. What this shows is the average seed production per meter squared, or half meter squared, because that's the size of our quadrats, over the 11 years that we've been censusing this, with warmer colors, red being higher levels of seed set, and bluer colors being lower seed set rates. And what it should show is that it's blue both at this range edge and blue at this range edge, and there's a tendency to be highest in the center. So seed production rates do appear to be highest in the center, conforming with the prediction that populations in the center should be doing the best. Sorry about that, you'll have to believe my word for it. We have never seen an actual total extinction of a population, so we've never seen a population disappear completely, although in any given year a population may fail to reproduce completely. Either there's very little emergence, or whatever emerges in seedlings dies before they set fruit. And so for these 35 populations, more or less, that we've followed over 11 years, we've had 54 site year combinations in which a population has failed to reproduce. Let's hope that there's the best explanatory variables on whether a population goes quasi-extinct is whether it's south-facing or not. So this shows the aspect of the slope along which the population resides, and the more south-facing, the higher the extinction risk. It's also related to the population size or area of occupancy. Smaller populations are more likely to experience reproductive failure. But once you account for these variables, what we find is that there is, in fact, populations at both range edges, east and west, have a somewhat higher probability of extinction. So we've looked also at 20 of those 35 populations. We've looked at them more carefully and actually estimated demographic parameters. So every year what we do is we come in the winter and record in permanent plots how many seedlings come up in each of the plots. Then we return there at fruiting time and ask how many of them actually made it to fruit. We sample fruits elsewhere at the site to get a sense of how many seeds would be produced for each fruit. And then we've done that now for 11 years, but we also tried to get some sense of what the seeds did. So for 3 years, what we did was we put known numbers of seeds in little mesh bags that we buried. We'd come back, we'd place the bags in the field in October before the winter rains come. We'd come back in February, open up the bags, count the seedlings, remove them, put the bags back into the field, and then retrieve a subset of those bags 1 year later, 2 years later, 3 years later to get and test the seeds that we recover after that time, those varying time periods, and test them for viability so that we could get a measure of how long seeds last. And using essentially these estimates of both above ground vital rates, seedling survivals, fruit set, seed set, and below ground vital rates, we can create a matrix model of these populations and project what the population growth rate is. For the first 6 years of our study, or 6 or 7 years, we found a very strong declining relationship between easting, so western populations had higher stochastic lambdas compared to eastern populations. And then we were hit by the drought, or Clarkia was hit by the drought. And now what we find, so this is pre-drought on the left-hand side and post-drought. And while there is still a significant there is still a decline in population growth rate as you move towards the east, it's no longer significant. And it seems that in these last drought years, what happened is these western populations were among the hardest hit, and that has sort of forced down the relationship. So in terms of summarizing the demographic limits, we don't have any evidence that population occupancy is smaller at the eastern range edge. We do find evidence that seed production is lowest at the range edge and highest at the center. And then there are more quasi-extinctions at the range edge. And there's some tendency for population growth rates to be lowest, at least at the eastern range edge. So for the last category of explanations for range limits, we wanted to look at the evolutionary limits on range edges. And we first asked, are central populations better adapted to their environment than edge populations? Edge populations might not be as well adapted either because they lack genetic variation or they're getting inputs from gene flow from central populations. We also ask questions about how much genetic variation do these populations have? Are edge populations less genetically variable than central populations? And finally, we tried to estimate levels of gene flow using molecular markers. So the approach that we took for this work was we took 6 populations, 2 in the center of the Sierra, the portion of the range that is in the Sierras, 2 at the eastern range edge, and then 2 populations that were intermediate between center and edge. 3 of these populations were north of the Kern River Valley, and 3 were south of the Kern River Valley. And we used reciprocal transplant experiments to test for local adaptation. That's sort of the gold standard of trying to test for local adaptation. You ask whether populations do best at home relative to being away. But transplant experiments are actually also useful if you move plants beyond their current range limits to sites beyond the range edge. You can ask, can populations persist beyond the range edge? And if they can, that would suggest that there's been dispersal limitation, that they haven't made it to all the environments that are favorable. So we did a transplant experiment where we not only transplanted populations between sites within the range, but we also moved them beyond the range edge. And then we also collected plant material from these populations and created plant material, seeds for these populations, in which we could measure molecular variation and also quantitative trait variation. So this was the design. We have these 6 populations, 2 central, 2 edge, and 2 intermediate, 3 on the south side of the river and 3 on the north side of the river. And for the transplant work, they were only transplanted within a transect, so not between north and south. It was just too much work to do everything. So each population was transplanted to every site along its transect. In order to quantify genetic variation, quantitative genetic variation, we needed to create families of seed so that we could ask whether individuals from the same family were more phenotypically similar than random individuals from different families. And to do that, we did a crossing design. We generated all of our seed in the greenhouse. And what we did was for each population, I'm showing here just for one of the central populations, we created 24 pairs of plants that were derived from different mothers in the field. For each pair of plants, we reciprocally crossed them many times to generate lots and lots and lots of seed. And each family then, that created 24 full SIP families of seed, and each of those families was transplanted to each of the transplant sites. Does that sort of make sense? And then we did that same thing for the intermediate population and for the edge population. This took us 3 years just to create the seed. And then we planted them in the field, and what we use for planting is, because we need to keep track of which seed came from where, we use, these are fluorescent lighting grids that we cut up and transport by the hundreds out into the field. We affix them right to the surface of the soil and pin them down with metal hoops. We don't disturb the vegetation. And then we plant every other cell with seed from a known source. And that's what it looks like when we're doing our planting design. And then this is what a whole site looks like when from a distance. It's hard to tell, but each one of these little white dots here is a grid. And so that's one of our transplant sites, and we had 8 of them. It meant planting 37,500 grid cells and 150,000 seeds. So it was quite an endeavor. And that's why I couldn't have done this work without the help of lots of other people. What we do then is that we can record germination. Here are seedlings in each of the cells that are planted. We record germinants. We then follow them to see if they survive to flowering. We collect every fruit and count every seed, and so we get a measure of fitness for each plant that makes it through the whole process. And all of that, those data boil down to one single slide. This shows for each of the source populations, so center populations are in black, the intermediate is a green square, and the triangle is the edge population. What is the seed produced, the average number of seeds produced per seed planted of each of these populations at each of the transplant sites? And fortunately for us, the results were actually very similar across both transects. So I'll go walk you through what these results say, looking at just one of the transects. So first we can look at the center population, and what do we see? We find that the center population does better at its home site in the center compared to every other site that it was transplanted in, and it does better at its home site than any of the other populations that were transplanted there. So it expresses both aspects of local adaptation. For the intermediate population, it actually does best at its home site relative to the other sites. So there's some part of it that is locally adapted, but it's no better at its home site compared to the other populations. And then finally, the edge populations are real duds. They not only do they not do best at their home site, they actually always did better at the intermediate site, but they also don't do better than anyone else at their site. There was one difference between the two transects, and that was that on the northern transect, our beyond edge site was actually better than the edge site for all of the populations, suggesting that there is maybe some historical limit, there is some dispersal limitation, or at least in some years. What we didn't find, and what we had sort of expected, was that the edge population would do better beyond the edge compared to the center or intermediate populations, just because it's closer already to those environments than the other populations, and we didn't find that. So then turning to levels of genetic variation, for the molecular variation, we looked at levels of genetic variation at microsatellite loci and also at 9 nuclear loci. And then for the quantitative trait data, we measured both life history components, the germination, survival, final seed set, and also 4 phenotypic traits. I'll just show you data on lifetime fitness. So for the molecular variation, on the left here is the microsatellite variation, and here is the sequence variation. And fairly consistently for both types of molecular markers, there was less genetic variation in edge populations compared to center populations. But for the most part, the reason why there was less variation is that the edge populations had fewer private alleles that were exclusive to those populations, and they were missing some of the private alleles that were present in the center populations. And if we do further analysis of the data, there's even some indication that the center populations had experienced a fairly recent increase in abundance, but there's no evidence of edge populations having experienced a real bottleneck. when we look at levels of quantitative genetic variation, and this is sort of the same kind of graph as the transplant data, we looked at, this is plotting genetic variance on the y-axis as a function of transplant site on the x-axis for the three different source populations. And in no case did we find that there was an effect of source population on the amount of genetic variation expressed for any of the traits that we looked at. And if anything, there was sometimes an effect of transplant site on the expression of genetic variation, but when that was present, it tended to be highest for edge sites or beyond edge sites, not for center sites. So this wasn't what we had expected. We had expected to find either less genetic variation in edge populations or in edge environments, and if anything, we found the reverse. If we use the molecular data to estimate gene flow rates, we do find that gene flow is biased from center to edge. This parameter M is proportional to the contribution that migrants make to the gene pool of the recipient population. And if you compare the migration rate from west to east, it's about double the migration rate from east to west. So in summary, for the evolutionary limits, we have strong evidence that center populations are locally adapted, but edge populations are not. There's at least occasionally in this particular year, the beyond edge, one of the beyond edge sites was suitable. We do find lower levels of molecular variation, but we don't find any evidence that quantitative genetic variation is lacking. But we do have some evidence that gene flow is biased from center to edge. So now we're actually testing whether this gene flow is maladaptive. We've created hybrids between center and edge plants and back crosses, and we're analyzing another transplant experiment that planted these hybrids, parentals, and back crosses at each of the sites to ask, how does gene flow affect performance? So to conclude, we do find some support for many of the hypotheses on range limits. The environment, the abiotic environment, does appear to deteriorate towards the eastern range edge. This is reflected in lower population growth rates and lower productivity, potentially at both edges. The level of population adaptation does vary geographically according to some of the predictions, that with center populations being well adapted, but edge populations not. But the limits on adaptive evolution may not be due to low genetic variation at the range edge, but rather to gene flow from center to edge. So in the future, what are we doing? We're continuing our long-term demographic work, and in particular, we're restarting the seed bank studies to get more estimates of below-ground vital rates. Our data also, we now have enough data over enough years of different climate and also varying density to ask how are vital rates varying with climate variables and with density dependence. Our demographic data also indicate to us that different populations are more or less sensitive to changes in immediate germination versus delayed germination. So we're trying to get at that by reciprocally transplanting populations that are demographically similar in terms of their sensitivity or demographically dissimilar. And then finally, we're doing the test of whether gene flow matters to adaptation by transplanting hybrids back crosses and parentals into center and edge environments. And that is, we had a lot of help, not only from my collaborators, but I roped in a number of graduate students who were usually doing something entirely different, and postdocs to come help on occasion with our censuses or our transplants. And then we've had a lot of help from a large number of undergraduates that we brought out into the field. And that, I'll take questions.
This is a comment or a question, but I was involved in a very large-scale reciprocal transplant experiment years ago across Europe, where in this case the central population was Switzerland and the range edges were Sweden, and Greece. And we found a very similar pattern in general in terms of adaptation, where the central populations did better than anybody else in the center, crashed as you went toward the edge. The edge populations did best at their edges, but did worse than everybody else.
Okay.
They didn't do best there. They did really well in Switzerland as well. It's sweet stuff for everybody, but not as well as the central populations and so on and so forth. So I'm just curious, is that a general pattern that you've seen more often? This is just a general trade-off between intolerance and massacre.
Well, yes, and what I think in our particular case is that the edge plants are genetic. I showed you the trend where plant height is smaller as you get towards the range edge. That's both an environmental effect and a genetic effect. So you move the edge plants into the center, they grow bigger there, and so they're more productive, they have more flowers and fruits, but they can never get as big as the central plants. Whereas everybody collapses down to the same size in the marginal environments at the edge. Now, is it the case that in a very bad year, in a very dry year, that's when the edge plants would have an advantage? There's some kind of trade-off between being able to get big and being able to tolerate really stressful conditions. I don't know. We only did this in 1 year. We have done other transplant experiments and have never found the edge crop in 2 other years, and found similar results. So we've never found a year in which the edge plants do better than the center plants at the edge, but it could have just been that we didn't challenge them with a hard enough year. But yeah, it's interesting.
And the situation I'm talking about, when we hit winter, nasty winter, everybody keeled over except for the Swedish and the Irish plants. And same thing in Greece, except it was hot.
Right.
Everybody keels
Right.
over. But the general pattern is right.
Yeah, no, it is kind of interesting. I think there is something about in more productive environments, plants are genetically bigger and they can take advantage of that, but the plants from edge environments are genetically constitutively smaller or less productive, yeah.
It seems to me that the same approach might be used for looking at species at risk, and sort of a reverse process. But in species at risk and rare plants in the northern regions, where there's both bird and mammal herbivores, that they have to be factored into the model.
Right.
Because they fertilize, but they also are selective.
Mm-hmm.
And they may be selective only seasonally,
Okay,
when the seed, for example, are available.
right, yeah,
And then they may be seed dispersers.
uh-huh.
But on the other
Or they
hand,
could
they're
be
also
seed predators.
preventing the rare plant in the site from successfully reproducing.
Reproducing, yeah. We find, I mean, I've showed you the pattern where plant herbivory declines as you go towards the eastern range edge. But one thing that we've discovered is when we move those plants beyond the range edge, they actually stay greener for longer than any plant naturally beyond the range edge. And so they're the only green thing around. Clarkia, the genus, is called Farewell to Spring because it's the latest thing to flower. And so they're green beyond the normal time. when you move them beyond the eastern range edge. And then they can get very palatable to rabbits and ground squirrels. And the biggest cause of mortality beyond the range edge was not drought, but being chewed, completely chewed. So yeah, mammals can certainly affect distributions. Yeah.
So the quick limiting factor might be whether the herbivores are threatened by climate change.
Right.
And
Right.
the environment is changing,
Right.
not because of that plant.
Right.
Plant is part of the sustainable.
Right. Yeah. Yeah. Interesting.
Have you ever compared plants on these?
Yes, we have a paper that's in production, or hopefully in production soon, where we reciprocally transplant. We haven't looked at mammal herbivory. One of Dave Muller's students is looking at that, but we did transplant fruiting stems from center to edge, between center and edge environments, and looked at how much they got hit by herbivores, the fruit herbivores, and what we found was that herbivory rates were higher in the center, but they were highest on edge plants. So they seem to be less well defended, yeah, which is intriguing, but we don't know why.
Do you know what those defenses are?
Well, let's see. I don't know if we have we think they are anthocyanins. So the plant is, if you remember the picture of the flower, they have spots on the flowers, and we wondered whether that was attractive to pollinators, because there's variation in spotting frequency, and you tend to get less spotting as you move towards the eastern range edge. But the only effect we found no consistent effect on pollinators, but we found that unspotted plants are more susceptible to fruit herbivores. So something about potentially the anthocyanins that are in the petals are also in the fruits. And spotted plants then have more anthocyanins in the fruits. So, but we haven't taken it beyond that, yeah. And there's some evidence of a trade-off. The most spotted plants produce fewer flowers. So anyway, but yeah.
Do you expect to see any effects of the recent surge in precipitation?
Yes, well, I was just out there in February because we had another experiment, this transplanting demographically similar and dissimilar populations. And they're just carpets of Clarkia. So I'm actually, because we've seen a tenfold reduction in abundance over these past 4 years of drought. So clearly they are, hopefully they will recover this year and produce a good seed crop this year. So yeah, I'm hoping. Last year, it was an El Nino year, which typically brings moisture to California, but it only reached the northern part of California, not the southern part. So we didn't see the effect where we were. Yep.
So, first, easy question is like, so when you go east, like, you showed like the pollinator limitations,
Mm-hmm.
but the pollen wasn't limited, right? Is that because of the mating system? Are they self-limited?
There is a selfing population subspecies that does occur to the east of where we work, and it is self-pollinating. And what came first? I'll show you. So sometimes I feel like I'm asking the wrong question, because we do have a Clarkia that evolved to occupy the eastern region. It's the sister species of the one that we work on, and ours, the one that we work on, is cross-pollinating, and the species that occurs to the east, they're called subspecies, but they might as well be species, is self-pollinating. and it's able to self-pollinate because the anthers and stigma mature at the same time and are right on top of each other. We know from molecular work that, so this is doing molecular analysis of outcrossing and selfing individuals from a bunch of different populations, that this happened only once, or at least there's only one successful evolution of selfing. So the selfers form a monophyletic group. It's also apparent when we do phylogeography within each species that this zone of contact here is not where the speciation event occurred. This is a zone of secondary contact. So the Parviflora evolved somewhere down here, as best as we can tell, moved up the eastern side and out towards the west, and now they're facing each other. So it can happen, but it happens relatively rarely. Obviously, it's only happened once in this species. Yeah, but it seems to be able to do better there because it's not only self-pollinating; it flowers earlier. So it's not chewed down by rabbits by being the only thing green out there. So it's evolved a whole lot of differences to occupy the eastern side.
Any last question?
So, how do you interpret your amazing amount of data about the genetic variation? And why do they express more genetic variation of the range?
Why? I don't know why it expresses more. There was one trait that showed the opposite trend, and that was genetic variation in flowering time. And there, the expression of genetic variation was lowest at the edge and beyond the edge. But there was no effect of source population on the expression. So, some people have, there have been arguments in the literature both for and against harsh environments increasing the expression of genetic variation or tamping down on the expression of variation. And in our case, it seems to be that there's an increase in the expression of genetic variation. I don't know what the mechanism is, but yeah.
Any last questions?
Thank you.
All right, thank you, Monica. Especially have our famous speaker.
Oh my gosh. Thank you.
That's a great talk.